Welcome to today's webinar. I thank you all for joining. The presentation that I will be giving today is titled De-risking Early-Stage Clinical Trials, an Upstream Continuous Production Solution for Biologics. As we all know, the cost of commercializing a drug is very high, and it continues to increase. For example, last year's data showed that it costs approximately $2.6 billion in aggregate costs to commercialize a drug. This is approximately a threefold increase in just the past 12 years. In addition, the rate of failure is also a very high compounding risk. Close to 1 in 20 drugs for oncology applications do make it to market. But on the flip side, that's a 95% failure rate. For small to mid-sized biopharma organizations, this high risk of failure leads to a considerable reluctance to commit to resources to sustain the development of novel therapeutic proteins. If you could get 3 molecules into the clinic for the price of 2, well, then you've just reduced your risk of failure. This presentation provides a solution to help you reduce your upstream manufacturing costs. Currently, the biomanufacturing platforms typically used are the large-scale fed batch stirred tank systems. They're very cost-effective at making hundreds of kilograms of protein utilizing the CHO platform, with tighter ranges in the grams per liter range. But these platforms are not the most cost-effective when it comes to sub-kilogram needs, which are typically associated with toxicology studies, animal studies, ex vivo applications, and the all-too-familiar, difficult-to-express proteins. The cell culture company uses the Acusys Perfusion Bioreactor system in our CDMO services to help manufacture cost-effectively these sub-kilogram protein quantities. How can this technology help you reduce manufacturing costs? Well, one of the first areas is that we have absolutely no seed train, or virtually no seed train with this technology. These bioreactor systems typically utilize a 5-liter shaker flask to help inoculate the equivalency of a 2,000-liter tank. In this pictorial here, you'll see that the cells that have been inoculated into a perfusion hollow fiber bioreactor take approximately 2 weeks to get to the point of confluency. In a sense, the scale-up takes place within the same bioreactor system. This lack of seed train translates into pricing that is approximately 20-fold less than traditional fed batch single-use disposable systems. The graph here shows that there's approximately a $35,000 cost in labor and raw materials to get to a 2,000-liter single-use disposable bioreactor. With a similar size platform in the hollow fiber, you can see that these costs are more in line with only around $2,000. The next advantage of this technology is its footprint and utility requirements. The Acusys Accelerator single-use disposable system does manufacture in a GMP-compliant format. It can provide both pilot and production scale capabilities. It is fully automated and greatly reduces the technician time. It's a closed system, so it can be used in a multi-product facility. It's equivalent to approximately a 2,000-liter stainless steel or single-use disposable bioreactor system. It is a single-use disposable and has been since 1985 when this technology hit the market, and the utility requirements are also relatively simple. It requires a 120-volt power source and uses a 100% CO2 gassing supply. There is also a requirement to have either an Ethernet or network connection. Last but not least, the size of this system fits through a standard-sized door and is on wheels. This slide shows what the labor and raw material costs would be to produce approximately 200 grams of a human IgG. The bars on the far left are for the accelerator. The bars in the middle are for the stainless steel tank. The bars on the far right are for the single-use disposable. The take-home message here is that with these types of expression platforms, you're looking at approximately a 20 to 40% reduction in labor and material costs for the perfusion hollow fiber bioreactor when compared to the stainless steel and single-use disposable tanks. The third advantage of this technology is its ability to maintain steady-state metabolics and protein production. The slide here shows you 2 different cell lines that were cultured in a range from approximately 70 to 80 days and exhibits the stable pH level that was maintained for this culture for that entire length of time. This slide here shows in a similar manner the glucose levels and lactate levels for these same two cell lines during that time period. Take note at the very beginning you have the traditional increase in lactic acid in a very short period of time, and then the traditional decrease in glucose. Then maintaining these set points is something that these automated bioreactor systems can help you facilitate, so that you can find, in a sense, your cell line's sweet spot as to where they best produce the protein of interest. The last slide in this scenario shows that you also have systems that are capable of producing protein for long periods of time. The take-home message related to the advantage of stable metabolics and protein production is that your protein's manufacturing process will not be determined by cell line stability, but more by economic factors such as how much protein do you need annually and what is the protein's shelf life. These two factors will help you determine the length of your manufacturing process, not your cell line's stability. Our Perfusion bioreactor technology is used not only in our CDMO manufacturing, but is also available to the market. This technology has been single-use, as I mentioned earlier, since the mid-1980s, and offers a range of scalable bioreactor systems from both pilot to commercial. The key to the scalability advantage is found in the utilization of the same size cartridge for all of these systems. These cartridges maintain the same form, fit, and function, and are connected in parallel. This format allows for a significant reduction in validation costs as your process scales up. As seen in this slide, the scalability of the technology is focused at what you will see in the y-axis, with units of how many milligrams of protein can be produced per day per cartridge. Take note on the x-axis that the first set of bars are from a 1-cartridge system, the second set of bars are from a 2-cartridge system, and the third set of bars are from a 10-cartridge system. Once again, cell lines A and B, you will see that there is a linear manufacturing no matter how many cartridges you use. This is the data that we believe will help support the reduction in validation costs as your manufacturing process does scale up to meet the needs of commercialization, which is what we all strive for. As a result of this technology, you can see from the data on these next two slides that difficult to express proteins can also be manufactured to generate more protein per unit of media consumed, and also in a smaller volume. The membrane technology allows for the culture and concentration of the protein of interest to be performed concurrently. In addition, the collected supernatant is removed continuously and comes out in the form of cell-free supernatant. This fifth advantage also helps reduce downstream clarification and manufacturing costs. For this TNF Beta FC fusion protein, you'll see that based on the consumption of 400 liters of media, whether it was in a fed batch or a hollow fiber, that the hollow fiber perfusion bioreactor was able to manufacture approximately 6-fold more protein when compared to the standard fed batch technology. In addition, all of the protein of interest was found in approximately 5 liters and not in the overall volume consumed, which would have been in a batch fed of 400 liters. This is in reference to what I had communicated earlier in helping to reduce downstream processing costs. This slide here, once again, exhibits the data regarding another FC fusion protein. If you look on the far right column, the hollow fiber perfusion bioreactor, utilizing the same volumes of media, were able to produce approximately 21-fold more protein and approximately 50-fold less in volume, once again helping to reduce your downstream processing costs. Now, if you have a full understanding of fed batch stirred tank technology and are not familiar with hollow fiber perfusion bioreactors, then you may find it difficult to compare and contrast their features. This table shows the information in a concise format. Take note that the green colors actually show that these two technologies do have a lot of similarities. They both support mammalian cell culture expression systems. They're both single-use, and they're both closed systems. Also, each having been on the market for greater than nearly 30 years. But the differences that we point out here between these two systems is where they will help you reduce your manufacturing costs. The system can help you maintain a homeostatic environment for many days, helping to produce more protein, and you can concentrate your material concurrently while culturing your cells to help reduce additional downstream processing costs. This technology will also help provide linear scale-up and does not require a seed tray. Although many of you may not be aware of this, Perfusion hollow fiber bioreactors have been used in the market for over 35 years. Originally patented to support mammalian culture in 1974, they're still in use today globally, supporting manufacturing for in vitro diagnostics, clinical trial manufacturing, and is used for an FDA-approved biologic. The highly vascularized network of hollow fibers allows for distribution of nutrients and removal of waste in a manner that is nearly identical to the tissue and the capillary network that makes up all mammalian tissue. A closed view via electron microscopy shows at the close proximity of the cells to the fibers. A closer EM photo shows the pores in the fiber as seen in the lower right corner. In addition, the cell mass shows a highly porous yet compact cell density, allowing for flow of nutrients, waste, and soluble proteins in and around the cells so as to maintain their vitality. A question we often get asked is how homogeneous is the cell mass inside the fiber bundle? Well, these NMR photos show the cell mass presented as white in the image. Take note of the progression of the mammalian cell culture from day 1 to 5, where you see equal distribution throughout the fiber bundle. Thus, this allows for maximum use of the available cell culture space for culture support and protein production. This slide provides an overview of clinical and commercial drugs that have been manufactured using Perfusion hollow fiber bioreactors. Perfusion hollow fiber bioreactors are a suitable alternative to fed batch tanks to support clinical and commercial manufacturing of biologics. In conclusion, Perfusion hollow fiber bioreactor systems can help you reduce upstream manufacturing costs by virtually eliminating the seed train, reducing the size of expensive cleanroom facility space and utility needs, by providing a linear, scalable platform to reduce validation costs, and to significantly reduce downstream clarification and concentration process steps, as these unit process operations are fully integrated into the Perfusion bioreactor process. I thank you today for your attention and participation in the C3 webinar.